Thank you so much, Roberta, and thank you so much to the organizers for inviting me to this beautiful place and giving me the opportunity to speak at this fantastic conference. What are condensates and why should you care about them? Well, this is crystal structure of HNR and PA1, or at least of the folded domains of HNR and PA1. HNR and PA1 is a protein that helps in alternative transcription, alternative splicing, is also found in the cytoplasm and goes into stress granules, has lots of interesting functions in cells, and is also strongly related to disease processes, and I'll show you a little bit about this later. But just looking at this X-ray structure, we're missing quite a few things. I have the impression that it gets really loud. I don't know whether the sound is good back there, whether I should speak loudly or not. Okay, good. So it has a long, intrinsically disordered region. We call this actually a low complexity domain because it doesn't use the full alphabet of amino acids. And we've already heard quite a bit about IDPs yesterday from Martin and from in some short talks. But here we are particularly interested in the fact that this IDR can mediate phase separation. I need to switch this around to be able to show this. And this IDR alone mediates phase separation of this protein. So what's phase separation? Phase separation crystallographers see this a lot, of course, in their crystallization trials. And this is not what you want, but it can be beneficial for growing crystals, right? But this is often under very non-physiological conditions. But nature has taken advantage of this principle to enable phase separation of proteins and other macromolecules under physiological conditions. And this is enabled by multivalent interactions that can make a three-dimensional network. Here, this is from a famous example from Mike Rosen's lab, where there's a protein with repeats of SH3 domains that binds to another protein with repeats of proline-rich motifs. Each of these can bind to the SH3 domains, and then you get large non-covalently cross-linked complexes. And if these have low enough solubility, then they can form a separate phase here. And this is now a phase-separated condensate. Just the multivalent interactions are not enough because you can make this percolated network at high enough concentration. You also need this relatively low solubility and couple this percolation to a density transition to get these condensates. This happens in cells. And here first you'll see these condensates. They have fluid properties in vitro, one protein in solution. But we also see these kinds of behaviors in cells. This is cells that are stressed with arsenide stress, and you see stress granules forming. And you see that these are dynamic entities that can fuse and coalesce. And in a little bit, the stress is going to be washed out, and they will disassemble again. This is not just one protein. This is a whole collection of RNA-binding proteins, specifically G3BP, which is labeled here. It's RNA, lots of RNA-binding proteins, including HNR and PA1. So they're not the same, but we can understand the formation of these stress granules via phase separation coupled to percolation kind of processes. There are lots of these types of bodies in the cell. Really important example that you all know from textbooks are nucleoli. There are nuclear speckles, P-bodies, and stress granules in the cytoplasm, and there are really dozens of these kinds of bodies. And they've, of course, been studied by cell biology for a long time. We have inventory lists of things that are within them and so on, and have an idea about their biological function. But what was kind of missing was how they're being formed. And this is an important and interesting question, because if you think about it, these are not just very large complexes, right? These are on the order of hundreds of nanometers to micrometer-sized assemblies. They are not enclosed by a membrane, but they can concentrate specific components within them. And how do they do this? If you had to build such a large assembly just by typical protein-protein RNA interactions with typical affinities, you need very high affinities to build such a large assemblies. And that would make it rock solid. But I showed you that these are very dynamic assemblies, right? And you can do fluorescence recovery after photo-bleaching on them and so on, and you see that components enter and leave these assemblies. So they are phase-separated condensates, and the idea is that their properties can help us understand their biology. There are not only these types of bodies that are being formed by phase separation, but other processes in the cell that are mediated by phase separation as well, as has become clear over the last few years. For example, heterochromatin formation, the function of superenhancers, and perhaps other functions in transcription, RNA metabolism in P-bodies, membrane receptor clustering and the downstream signaling processes, and even processes that involve the formation of new membranes, like autophagy and endocytosis, are mediated to some extent by phase separation. And then we have the nuclear pore complex, and the selectivity filter within it is mediated by these kinds of percolated interactions that we're going to talk about. And this has really changed how we think about compartmentalization in cells. And, you know, one could say that perhaps we're going into an era of cell biology 2.0 with this. So how does phase separation work, right? I briefly told you about these multivalent interactions, multiple domains interacting with a binding partner with multiple motifs. What we understand about this quite clearly is that the valence of these two proteins for each other is really important for the driving force for phase separation, which can be quantified in these kinds of phase diagrams here. This is a schematic phase diagram that I just drew, but one can measure these, right? Where if we titrate protein B versus protein A here, we can see that out here, outside of these lines, we would have a single solution, one phase in our system. And once we cross these lines, we'd have two phases. So there would be droplets present in the sample. And there would be coexisting dilute and dense phases. And you can see that for a valence of 3, the gray line, you need higher combined concentrations of A and B to make this happen and to get droplets. And if you have a valence of 4 or 5, you need a lower concentration. So the valence and the interaction strength of these different motifs for each other really determine the driving force for phase separation. So this is pretty well understood. But how is this for intrinsically disordered regions? And I showed you that this intrinsically disordered region of HNR and PA1 can undergo phase separation by itself. Right? So there's some cropping problems from transformation to PC. But basically here you see the sequence of the low complexity domain. You can see that this is really high and small polar residues. Lysine and serine make up nearly 50% of the sequence. And then we have a few aromatic residues uniformly spaced throughout the sequence and a few charged residues. And we have been wondering, or we were wondering at the time, how phase separation is encoded in this sequence. And by the way, we call this a prion-like low complexity domain because of its sequence properties. And it shares this with lots of other RNA binding proteins. And mutations in this region can lead to familial forms of neurodegenerative diseases, such as ALS and frontotemporal dementia. And these types of domains get translocated in certain forms of cancers and result in fusion oncoproteins with DNA binding domains, and then can drive leukemias, for example. So these types of domains are quite interesting, and their phase behavior seems to be important to understand biology and disease processes. So to look for the interactions that really drive this self-association, we took the stance that we said, okay, in dilute solution, these motifs should also lead to compaction of the protein and to self-interactions in the protein. So we used NMR spectroscopy to look which types of residues in the sequence really like to mediate self-interactions. And this really pointed to aromatic residues without me showing you the data for this. And we used small angle X-ray scattering to get high quality data and to see that this protein was really quite compact. And that gave rise to the hypothesis that it was really the aromatic residues that compacted the protein and therefore were probably important for its phase separation. And then to test this, we titrated the number of aromatic residues in the sequence, enhanced it by 1/3 or reduced 1/3 or 2/3 of the number of aromatic residues, and that had a strong effect on the single chain dimensions. So increasing the number of aromatic residues compacted the protein more, really showing that the aromatic residues were adhesive elements in the sequence, and we called these sticker residues. And the spacers are intervening residues that probably mediate lower interactions and can also determine the solubility of the protein, and therefore, of course, also contribute to the phase separation properties. But based on our experimental data, where we did these titrations of these numbers of residues and measured the single chain dimensions, Rohit Pappu and Alex Holhouse, with whom we collaborated here, and we collaborated a lot with Rohit Pappu's lab overall on this topic, and you'll see this, they extracted pairwise interaction strength of sticker-sticker interactions, sticker-spacer interactions, and spacer-spacer interactions. And that allowed them to do coarse-grained or lattice simulations here. We put lots of chains of this in a box, and these are only encode stickers or spacers, the individual beads. So it's a very simple model. And then they can simulate the phase behavior, and you saw that there are dense phases here, and then there is dilute protein. And if one then does this as a function of concentration, one can extract these phase diagrams. And these are now phase diagrams of single protein and versus the temperature. And then we can see at a given temperature, we need a certain amount of protein to hit this coexistence line to first get droplets. And if we were, let's say, here at this concentration of error plus, the solution would decompose into a dilute phase with this concentration and a dense phase with this concentration. These simulated phase diagrams, we were able to compare them with our experimental data, shown here as triangles, and they fit really well. And this told us that, you know, these aromatic stickers were really the important, or were contributing a large amount of the interaction strength to drive phase separation. What it also shows us is that, indeed, the higher the sticker valence, so the more aromatic residues we have, the higher is the driving force for phase separation. Very much in agreement with these domain motif type proteins that I told you about before. We can, of course, also change the interaction strength, right? If we use different types of aromatic residues, they might have different interaction strength. And here, Anna and Wade made variants of the protein where they replaced tyrosines with phenylalanines or the other way around, so that we have then only one type of aromatic residues in the sequence. And you see here that tyrosine is a stronger sticker than phenylalanine because we need a lower driving force for phase separation. We need a lower concentration here to get to hit the coexistence line. We can also look at the spacer residues. Specifically, I told you there's a lot of glycines and serines in this sequence. And we can ask ourselves, you know, how little interaction do they actually contribute? And does the serine perhaps contribute a little bit more than the glycine? And that might be expected, given that it has the extra hydroxyl in the side chain. Or, on the other hand, because it's bulkier than the glycine, it might inhibit a little bit the interactions of the aromatic residues in the chain, and it might really like to interact more with the water, and therefore it could suppress the density transition. It could still make a network, but it could suppress the density transition to make the separate phase in a certain way. And so to test this, Anna and Wade replaced increasing number of glycine residues with serines, and this reduced the driving force for phase separation, as you can see, by increased saturation concentration. So we need more proteins to make droplets of these variants now. Or we can replace all 23 serines with glycines, and this reduced the saturation concentration. So now we need less protein. So this really shows that glycine really enables phase separation better than serine here. And we played similar games where we mutated serines to threonines and asparaginines to glutamines, and we always saw that the bulkier residues here reduced the driving force for phase separation. And we look at this as effective solvation volume. So how much does the chain like to interact with the solvent? Small residues have a small effective solvation volume. Larger residues, and definitely charged residues that really like to be solvated, have a large effective solvation volume and can therefore suppress the density transition. And then we have a number of stickers with different interaction strength that can make this networking. And the net charge per residue, I didn't show you this, but the net charge per residue has an important effect on solubility of the protein and therefore contributes to the driving force for phase separation. In these kinds of experiments and all of these variants that we looked at, we always also measured the single chain dimensions by small angle X-ray scattering. And Rohit and Nina, in this case, were able to extract these interaction strength, here pairwise interaction strength of stickers, and then also interaction strengths of some spacer residues, and can again do these, oops, that didn't play, these coarse-grained simulations, and we can extract phase diagrams, and the experiments and the simulations fit really well. So we think we really understand how phase separation is encoded in these sequences quite well. And there are, of course, other models by other people, including Preston and Larsen, who spoke yesterday, who have used this kind of data and lots of other data in the literature to extract models that are slightly different and have a slightly different framework, but that can also describe this type of phase behavior very well. So we can extend this framework, the stickers and spacers framework, to other types of sequences. We used it here to understand phase behavior of intrinsically disordered proteins. We talked about these linear multivalent proteins, where basically the whole domains are the stickers or the binding sites in the domains. But some folded proteins can also undergo phase separation, and they have some associative patches on their surface. So what does that all mean, and what kind of properties are the result of this kind of phase behavior? So the phase diagrams that I showed you had these straight tie lines, or these horizontal tie lines. That means that the dilute and the dense phase here were, of course, at the same temperature, right? If you generate condensates in your sample, the dilute and the dense phase will have the same temperature, right? But if you, let's say, change the ion concentration and looked at your phase diagram as a function of the ion concentration, there might not be a horizontal tie line here. The ion concentration in the dilute and the dense phase might be different, right? And it's actually to be expected that not only macromolecules may show unequal partitioning between dilute and dense phase. If you have charged macromolecules, that you might have uneven ion partitioning in dilute and dense phases. And so this has been basically ignored in the field, also because it's difficult to measure. So we teamed up with Rohit again to do some measurements on this. And basically we separate dilute and dense phase from each other. We can measure protein concentration by HPLC or simply by absorbance and the UV range. And then we can use inductive coupled plasma mass spectrometry or ion selective electrodes to measure the concentrations of ions in those dilute and dense phases. And we again did this on the low complexity domain of HNR and PA1. It is slightly positively charged. And we see here negative slopes of tie lines for sodium and positive slopes for chloride just at the very high concentration. They start becoming negative. Right? And what that means is that there are more sodium ions in the dilute phase than in the dense phase, and there's more chloride ions in the dense phase than in the dilute phase. And this makes total sense in terms of electron neutrality, right? Since the protein is positively charged, we better have some more chloride ions in the dense phase. But you see here at the higher salt concentrations, it's not actually that we just get a lot of chloride in the dense phase. This would result in unequal osmotic pressures in the dilute and dense phase. The osmotic pressures between dense and dilute phase need to be equalized for this to work overall. And so basically, this can limit the amount of protein or the driving force for phase separation, the amount of protein that goes into these dense phases. So there's some interesting interplay between ions and proteins in the formation of these. We can play around and change the charge on our proteins. And we have something that's even more positive, and of course we see the same, that we have sodium depleted and chloride partitioning into these dense phases. If we go to a protein version that has basically zero net charge, we see that there's relatively little incorporation of these ions into these dense phases. And then if we have a negatively charged protein, then we also see that chloride doesn't like to go into it anymore. Sodium also really doesn't like to go into it. So there is some really interesting regulation going on here. Well, if we have this unequal ion partitioning, that means we have electric potentials between these two phases, right? And basically the same equations that apply across membranes. And we can calculate them because we have measured these ion concentrations. And you see the potentials plotted here. The dominant potential is really the potential that tells us how much unequal charge is over this interface. And you can see that depending on in black here, and depending on the ion concentration, the salt concentration, we get potentials of 25 millivolts, right? And this isn't quite like a membrane potential, but we're also looking at a very specific protein here. And you can get quite interesting potentials. And we think that they probably have implications for function. The interface potentials, their size, are influenced by the macromolecular charge, as I showed you, by the ion type, I didn't show you this, and by the salt concentration. This dominant potential indicates how much charge the condensate can store. The condensates basically are capacitors, and they should be able to do work. And there isn't a lot of data on this yet. This is a very new direction. But there is recent work by Yifan Dai, who showed that the formation of condensates within bacterial cells actually changed the electrochemical properties of the cells and had an influence on transcription. So overall, we have, there's this initial repertoire of cellular functions that may be mediated by phase separation that colleagues and I put together a few years ago. And one of these is sensing of temperatures, but perhaps also of changes in ion concentration in cells. You can localize things by condensation in the cell. You can inactivate things by recruiting some components of a reaction or of a signaling process into a condensate, and thereby taking them away out of this process. You can activate reactions by nucleating them or by enhancing the concentration. Or you can generate force, and for that you need certain viscoelastic properties. And this is exactly what we would expect, that these condensates, because they are a network of molecules within these dense phases, we call these network fluids, that they have viscoelastic properties. And these viscoelastic properties should be encodable in the sequence, and they should be able to do work. And you have all probably all interacted with viscoelastic properties. You've probably had silly putty in your hands. It can, over long time scales, flow like a fluid, but if you interact with it on very brief time scales, it bounces like an elastic solid, right? Whether it behaves like a fluid or a solid has to do with the timescales of the rearrangements of the physical bonds within the material relative to the timescale on which you interact with it. And so we should be able to do this also with these condensates. If we change the interaction strength within the condensates and therefore the lifetime of these bonds, that should change their viscoelastic properties. And so we tested whether this was the case in collaboration with Priya Banerjee's lab. And Priya has really been a developer and has adapted methods from soft matter physics to really be able to measure material properties in condensates. And here we used passive microrheology with optical tweezers, where we trap a bead within a condensate, And then we follow its trajectory over time, and from that we can calculate an autocorrelation function and extract the storage and the loss modulus. So some real measurements of material properties. And these gives us information on the elastic and the viscous responses in these materials. And just to show you what this could look like, right? If you have an elastic solid, you would get an elastic response, and this should be constant as a function of frequency. If it's a very simple material, you could get a viscous liquid where you only get a viscous response, and so on. And there's this Maxwell viscoelastic liquid that has both a viscous and an elastic response and some sort of crossover that is related to this lifetime of these physical bonds within the material. And so again, some problems with cropping, but when we look at the experimental data, and here we are comparing different A1-LCD condensates with different sticker strengths, either the phenylalanine, which is the weakest sticker, tyrosine in intermediate strength, or tryptophan is the strongest sticker, and then we can look at these viscous and elastic responses, and we see, first of all, that these are viscoelastic Maxwell fluids. We have both of these types of, I think the laser pointer is giving up, we have both of the viscous and the elastic response. Their frequency dependence is as we would expect. And then for the strongest sticker, we get a crossover here. We start seeing the crossover also for this sticker. And here we don't see a crossover. It's probably at much higher frequencies. So we can really change the material properties of these condensates by changing the sequence. And nature probably took advantage of this. So condensates are viscoelastic fluids. Their material properties can probably influence their functions. And there is an example of a prion-like domain of this yeast protein SLA1 that has been shown to do work by invaginating the cell membrane without actin in endocytosis processes. And its viscoelastic properties there are important. Material properties can change as a function of the age of the condensate. And this has been linked to neurodegenerative diseases. Specifically here to stress granules. You see our stress granules again. They are very dynamic in the beginning, right? But over time, they are formed upon stress, and polysomes are disassembled, and RNA and RNA binding proteins together condense into these condensates. I have, my laser pointer is, it's coming back now. And then over time, they can become less dynamic states. This is said here that it's solid-like. I think we don't really know exactly what happens at this point, but certainly we know that they become less dynamic. And when we looked at our A1-LCD condensates, or I should also say, sorry, that this is related to disease processes. If we have RNA binding proteins that have disease mutants that lead to these neurodegenerative diseases, these processes are being sped up. And they result in the end in solid inclusions formed by these RNA binding proteins, and that are the pathophysiological hallmarks in patients later. So if we look at, this is one big A1-LCD condensate, and we have lots of beads in this condensate, and we're tracking their motion to see how their mean square displacement, this gives us information on their viscosities, and how these change over time. And they slow down over time. And we can quantify this here, and we can see that over 24 hours, here for this particular protein where we have tyrosines in the sequence, we don't get any changes to the mean square displacement. But when we replace an increasing number of glycines with serines, then we actually get some slowing down over 24 hours. And in this variant, these condensates are completely arrested after 3 hours. So we can manipulate the time scale of their aging by manipulating the sequence. This does not entail changes in thioflavin T fluorescence. So there are not fibrils being formed here. And we wanted to know what the final material state is, because this is very hard to determine. If you do FRAP, you see decreased recovery and so on, but you don't know what the material state is, whether it's still a viscoelastic solid, and this has been viscoelastic fluid, and this has been proposed in the literature, or whether it is really turning into a solid. And so Priya devised this creep test. We were trapping the speed here in this optical trap and moving it at a constant speed for a certain distance, and then let it go. And if we had a terminally viscous fluid, then we would expect that we could just let it go and it would stay in its new place. But if we have a terminally elastic response, a terminally elastic solid, then the bead would recoil to its original position. And so when we tried this here, we saw these fresh condensates at 0 hours. Both the non-aging and the aging one behaved like viscoelastic fluids, as we expected. The non-aging one still behaved like this at 24 hours, but the aging one showed a clear terminally elastic response. We couldn't even move the bead for the full 5 micrometers. After 1 micrometer, we actually lost the bead from the trap, and it recoiled back to its original position, really showing that this is a viscoelastic solid at the end. So this dynamical arrest results in increased, I didn't show you this, but it results in increased beta sheet content. We looked at this by FTIR. There is increased beta sheet content, although we don't see any fibrils forming. And this turns these viscoelastic fluids into non-fibrillar viscoelastic solids. So I'm out of time, so let me see. I'll just say that we can also look at fibril formation from condensates, because we think that this is probably the more important process in these aging diseases. We had seen previously that this disease mutant of HNRMPA1 can form these fibrils when condensates are present. And let me just say that we've seen that. I will stop. I'll just make this one point. Okay, thank you very much. And that we saw that indeed there is nucleation at the interfaces where we get thioflavin T lighting up at these interfaces over time, and these interfaces can mediate nucleation. And if we look at these thioflavin T curves as a function of protein concentration, and there is, where we have blue and white curves, there we are low concentrations below the saturation concentration, and then here in these broken, where we have the broken lines, we have condensates present. Then we can actually see that for the disease mutant, we get what looks like nucleation and growth kind of behavior. We know there's nucleation at the interfaces. But for wild-type protein, we actually see that these growth curves don't really speed up with concentration. So these slopes are not getting, here they get much higher with concentration. And here that's not the case. And what we concluded from that, without really going into the details, is that, okay, overall the interfaces of these condensates can enhance nucleation of fibrils. The interior of the condensates can actually suppress the fibril formation, and they can serve as sinks for soluble protein. We think stress granules may not actually be a problem in cells, that they could actually be protective, unless we have these kinds of decisive mutations that really stabilize fibrils. And we actually see that fibril growth happens in the dilute phase and not in these condensates. And we think that this is because we have a different molecular grammar, or partially overlapping but distinct molecular grammar, these sticker-sticker interactions that mediate the formation of condensates, but zipper interactions that mediate the formation of these amyloid-like beta sheets. Okay, and I'll jump over this and say condensates are long-lived but dynamic network fluids. The fact that they are long-lived can be used for function and can drive reactions that are really slow, for example. Condensates are viscoelastic and can suppress or enhance reactions by emergent properties. I didn't show you examples from our work where they can enhance reactions, but there's really nice work in the literature. And their interfaces are characterized by electric potentials and have the ability to promote reactions. For example, the fibril formation or the nucleation of fibrils that we have seen in our work. And with that, I'll thank people in the lab who have been involved in this work and wonderful collaborators, facilities that we've been using, of course funding, and I'd be happy to answer your questions. Thank you.